Madam.
Good morning, students. Welcome back to another class of Botany second year. Unit 1 Plant Physiology, Photosynthesis in Higher Plants. We have started this session and completed two parts. In the first part, the historical background of scientists who contributed for the understanding of photosynthesis was dealt. And in the second session, we have learned about the site of photosynthesis. What is all this photosynthesis taking place, where it is taking place, the structure of chloroplast, the dimorphism in the chloroplast was dealt. And assignment was also given accordingly. And some of the students, they have forwarded their work. And so I take the pleasure of appreciating them. R. Tanusri, M. Sneha, Rama Lakshmi from Viharabad, D. Sunita from Balanagar, R. Triveni from Vaira, Archana, Shivani, Harika, Aisha from Vangara, TSRJC. So all these children are doing their work and I appreciate their work and their neat diagrams. I hope all other children are also doing the same thing, and the thing you need is to forward it to your JLs, and JLs, please find out a best one or the quick response from your student, forward it to me so that I can appreciate them in the live session. So let us begin with today's topic. In today's topic, we are going to learn about light reaction. We have learned about site of photosynthesis, that is the chloroplast structure. Chloroplast structure is very, very important, and it is intact structure present in the plant cell only, and some of the photosynthetic bacteria. This chloroplast structure is the site for photosynthesis, which will be having specified location for conducting light reactions and dark reactions. As the name indicates, light reaction, that means sunlight is going to get harvested in this. So let us see what is this light reaction. It is a photochemical phase in the photosynthesis. In the photosynthesis activity, we will have photochemical phase where light will be harvested, and a biosynthetic phase, which is also considered as dark reaction, where carbon fixation will be taking place. So the energy required for preparing the food is nothing but the sunlight energy, the quantum energy, how it is getting harvested, how it is utilized for synthesizing ATP, NADPH, which are going to be further used in the dark reaction, will be dealt in today's topic. So you must be aware of each and every new terminology you are coming across in this session. And photochemical reaction, this photochemical phase is going to take place in the grana. Grana are nothing but the thylakoids which are located, piled up, and it is also called as thylakoid membrane. In the grana only we'll be having the chlorophyll pigment. Chlorophyll pigment, yesterday we have learned about chlorophyll pigment, its structure also. How this chlorophyll pigment, it is of 7 types, chlorophyll A, B, C, D, E, bacteriochlorophyll, and so this chlorophyll is very important for trapping the solar energy, and particularly chlorophyll A is very important, which is going to form the reaction center. So here in this light reaction, it includes first light absorption, water splitting the water. When water H2O is split, oxygen is getting released. Meanwhile, the hydrogen, protons and electrons are going to get separated. This is very important, and the protons and electrons which are liberated from these, the protons will be taken up by the electron acceptor NADP, and it is going to get reduced to NADPH. And the oxygen, it is a gas which is going to get released into the atmosphere, which is a byproduct of light reaction. And formation of high chemical intermediates, that is ATP and NADPH, is going to take place in this light reaction. So remember, what are the byproducts of light reaction means? First of all, oxygen is released, and here ATP and NADPH. These are the other two products which are formed by the end of light reaction. So here the raw material for light reaction to take place is sunlight energy, and the chlorophyll pigment is going to trap the solar energy, and the most important thing is splitting of this water molecule. So this water, sunlight, and the chlorophyll, these are the raw materials of light reactions. Sunlight energy is harvested by chlorophyll molecule, Meanwhile, it is going to split the water molecule and oxygen is liberated, which is a byproduct of light reaction, and some intermediates like ATP, NADPH are going to get formed. These ATP and NADPH are connecting link between light reaction and dark reaction, because light reaction products ATP and NADPH are utilized in the dark reaction. So remember these important points of light reaction. Now in detail, we are going to learn about light reaction. Here you can see in the next slide, light reaction involves the photolysis. As I said just now, water molecule H2O is going to split. How the water molecule is going to split? H2O. It is going to split into protons of hydrogen and electrons of hydrogen and oxygen will get separated. This oxygen, we can write half oxygen, or it is nascent oxygen. It is going to combine with another oxygen and they are going to become the oxygen gas getting liberated. So here for every one molecule of water, we'll get half oxygen. That means only one single nascent oxygen is going to form. And in photosynthesis, we are going to utilize 12 molecules of water. In the photochemical equation, just recall 12 molecules of water. We are going to use 6 molecules into protons. So this is first major thing which is going to take place. That means there will be a complex where the water molecule is going to break in the chloroplast, in the grana. So in the grana, remember in the chloroplast, inside this will be having the granum, thylakoid membrane. So inside this grana only the chlorophyll pigments are present and the water molecule is going to form photolysis complex. Photolysis complex means inside the grana. For example, this is grana. So in the inner membrane or inside, they'll be having the photolysis complex where water molecule is split and oxygen is released. And chlorophyll is also present here only in the grana. Chlorophyll, green color pigment. Okay. Now we'll move on. Next. Photophosphorylation. So this photophosphorylation is nothing but, photo means light, phosphorylation. Phosphorylation is nothing but addition of inorganic phosphate to ADP. ADP is adenosine diphosphate. So this ADP is going to get added up by ADP, adenosine diphosphate is going to get added up by inorganic phosphate and it is going to become ATP. ATP means adenosine triphosphate. So to the adenosine molecule, now 3 phosphates will be there. The terminal phosphate will be having the energy. Whenever ATP is nothing but the energy currency of any cell, energy currency of any cell. Okay, so this conversion of ADP by adding phosphate, inorganic phosphate, and making adenosine triphosphate is called phosphorylation. And this is occurring in the presence of sunlight. And so we have to write here photophosphorylation. Whenever we require energy, whenever any cell requires the energy, the terminal phosphate is utilized. And the terminal phosphate is utilized. And again, it will get back to this condition, ADP plus PI. Okay, so this is a thing happening related to photophosphorylation here in the granum thylakoid. So here in the grana, the synthesis of ATP from ADP and PI in presence of light with the chloroplast within the chloroplast is called photophosphorylation. It is of two types. So the most important and new concept you are learning here is two types of photophosphorylation. One is non-cyclic, another one is cyclic. Non-cyclic photophosphorylation and cyclic photophosphorylation. You are going to see in this in the coming topics. Here, before that, we will have a clarity about the chlorophyll pigment. What chlorophyll pigment is doing here? Quantasome. This term is given. Quantasome is a term given to the collection of the energy. Collection of pigment. It is nothing but a group of pigments molecules which are going to undertake the photosynthesis. That means they are going to trap the solar energy. The beginning of photosynthesis is by trapping the solar energy. So this pigment molecule has to trap the solar energy. And this collection of the pigments, these pigments which are going to trap the solar energy are located in the thylakoid, as you can see in the picture here. You can see this is the thylakoid, and inside you can see how they are arranged. They are going to make the antenna molecule. Antenna molecule is nothing but it is comprising of chlorophyll B, carotenoids, the accessory pigments are also there. And also we have the chlorophyll A. We also have the chlorophyll A in the center, and the chlorophyll A is going to be the boss here. That means whatever these antenna molecules are harvesting the solar energy, that means the accessory pigments, chlorophyll B, carotenoids, we will be having in the thylakoid only. These are also going to trap the solar energy and they will pass on. They will pass on it to the center chlorophyll A. This center will be having the chlorophyll A pigment, and surrounding this chlorophyll A pigment will be having the other pigments, accessory pigments. Okay, and these are going to make up the antenna complex, and this antenna complex is to trap the solar energy, and they are going to pass one by one. The solar energy will be taken and passed to the next, and finally chlorophyll A will be taking the energy, and it is going to be the reaction center. Always remember, chlorophyll A is going to make the reaction center. That means entire energy which is trapped by other accessory pigments is also given to the chlorophyll A. So here we will have the antenna molecules or antenna complex made by the carotenoids, chlorophyll B and other pigments, and they are going to give the energy to the chlorophyll A, and it is going to make the light harvesting complex. Light harvesting complex. And here you can see photosystem 1, photosystem 2. Just now I discussed about the light harvesting complex. It will make you clear what is photosystem 1, photosystem 2. Several complexes are involved in this process, light harvesting complex. So not only one light harvesting complex will be there. There will be several complexes all over the thylakoids. You know, imagine how many molecules can be there in the chloroplast. You will be having many number of grana thylakoid will be there. So in each and every grana thylakoid membrane, these complexes, light harvesting complexes will be there. Okay, their main work is harvesting sunlight. Okay, and here you can see the photon. Photon is nothing but the light energy packet. Okay, the quantum energy is there in the photon, in the sunlight packet. So here this photon is going to be taken by this antenna molecules, and here only one box will be there, chlorophyll A, that is going to make the reaction center. And when it will be taking the energy, sunlight energy, one a pair of electron will get excited due to energy intake, due to getting more energy. That chlorophyll A electron in the outermost shell, a pair, is going to jump. It will jump. When it will jump, it will rise high, and here there will be primary electron acceptor molecule will be present. It is going to take that one. So remember this light harvesting complex, where energy is trapped by the light harvesting complex, that is accessory pigments, chlorophyll B and carotenoids, and they are giving it to the reaction center. that is a chlorophyll A, and chlorophyll A, it is becoming a high energy state, and immediately two electrons, that means a pair of electrons are going to jump with that energy, and it is taken by primary electron acceptor molecule. So here, when it is taking place, we'll be having here photosystem 1 and photosystem 2. In short, it is called as PS1 and PS2 because they are named after the identification, the PS1 and PS2. This photosystem 1 and photosystem 2 is nothing but it is a place where the light energy is getting harvested. The wavelength P680 and P700. In short, PS1 is nothing but P700 and PS2 is nothing but P680. Okay, so P700, the red light area where the wavelength red color is trapped by the chlorophyll molecules or the light harvesting complex, it is going to make up our PS1 and P680. In short, P680 is nothing but photosystem 2. Photosystem 2, 680 is the wavelength. This is also red light area only, where red light is taken or trapped, and the light harvesting complex, they are trapping the solar energy, and here also the electrons are going to jump. So both are similar things only the thing, the wavelength. Okay, light wavelength will be P700 and P680, 680 nanometers of wavelength, 700 nanometers of wavelength is going to make the photosystem 1 and photosystem 2. These are named in the sequence of their discovery. And next, now it is clear that chloroplast having the grana, having the pigment molecules, and these pigment molecules are making the light harvesting complex where they are trapping the solar energy and becoming fully charged. Chlorophyll A molecule is going to make the reaction center. This chlorophyll A molecule, it will be losing its electron because of high energy intake, solar energy. A pair of electrons will become excited and jump. When they become excited and jump, electron acceptor will be there for accepting them. It will take, and that locations are PS1 and PS2. PS1, in short, it is P700 nanometer. That means photosystem 700 nanometer wavelength, and PS2 is at photosystem 680 nanometer wavelength. So here you can see this is the thing, how you are able to see here clearly. This is our sun, and it is giving the light energy, and the light energy is traveling, and when it is traveling, it is getting trapped by these accessory pigment molecules. These green colors molecules are accessory pigment molecules, and in the center we will be having the boss, and the boss is nothing but chlorophyll A. It is the reaction center. This reaction center may be located at two things, at P700 and P700 wavelength and P680 wavelength. Okay, so here we are having P700 means PS1 and P680 wavelength means PS2. So two photosystems will be there in the grana. Okay, I am mentioning here in the grana, both the photosystem 1 and 2 will be present. And you must know the importance of this photosystem. This photosystem is going to make phosphorylation. The phosphorylation incident to take place. That means ATP formation will be taking place, and also NADPH. NADP is going to get reduced to NADPH, and also photolysis, breakdown of water molecule. So it is happening here with the presence of PS1 and PS2 in the grana. And one more important thing you have to remember, there is only single PS1 in the stroma. Okay, when I am saying about in the stroma lamellae, stroma lamellae is nothing but the connecting link between uh, two granas. For example, these two are granas, and this is the stroma lamellae. So in the stroma lamellae, only PS1 will be there, Whereas in this grana, both PS1 and PS2 will be there. So this clarity must be there for you for a better understanding, so that you can understand about cyclic and non-cyclic photophosphorylation. So this is PS1 and PS2, two photosystem in the grana. Now I'll talk about PS2 here. In PS2, as you have seen, PS2 is nothing but it is located at P680 wavelength. 680 wavelength. When this RC means reaction center, okay, reaction center, and here when the chlorophyll molecules are taking the energy and giving it to the reaction center chlorophyll a pair of electrons are jumping here. When a pair of electrons are jumping, it is taken by here. Here you will be having the primary electron acceptor, which is going to accept it, and it is none other than plastoquinone. PQ stands for plastoquinone, and it will not absorb, it will be helping in transporting that electron. Okay, you can see here in PS2 clearly. Here you can see plastoquinone, cytochrome, okay, plastocyanin, and what you are seeing here, P700 you are seeing. P700 is none other than PS1. So here PS2, you are seeing that here PS2 is at P680. Reaction center is there, electron is jumping. When electron is jumping, electron transport system, this is called electron transport system, where you are having certain electron acceptor molecule like plastoquinone. You are having the cytochrome here, and plastocyanin is there, and immediately they are passing it to the reaction center P700, which is photosystem 1. So what is happening when the transport is taking place here from plastoquinone to cytochrome to plastocyanin? ATP formation synthesis will be taking place here. Okay, due to the energy getting liberated from that electron. That means you are seeing that it is going downhill. It is excited here and it is going downhill. And when it is going downhill, hill means Hill's reaction. Robert Hill discovered no on his name only. So when it here the energy is getting lost and it is going down and down and down, and it will reach the P700 photosystem 1. So this is called electron transport. So this is called electron transport. Okay, the electron which got excited in the reaction center of P680 will jump and will be taken by primary electron acceptor PEA, and it will be passed to the plastoquinone, cytochrome, plastocyanin. Okay, so these are going to be called as electron transport molecules. These are going to transport that electron and give it to P700, which is photosystem 1. Okay, this you be clear about photosystem 2. Okay, and further what happens in the PS1 we'll learn. Just I'll move on to next slide. I hope you are getting a clarity about electron transport here. How the excited electron are getting transport is going to make our electron transport system. So this is called non-cyclic photophosphorylation. Just now I said the ATP formation is taking place. ATP formation is nothing but phosphorylation. This phosphorylation is taking place in the presence of sun, photophosphorylation. What sort of photophosphorylation? It is non-cyclic. Now you are going to learn about the non-cyclic photophosphorylation. And for this you require two photosystem, PS1 and PS2. So in this light reaction, you are going to be familiar with non-cyclic photophosphorylation. Now that you are having a clarity about two photosystems, what are those two photosystems? The PS1. Where is the PS1 located? 680 wavelength. not PS1, PS2. PS2 located at 680 wavelength, and PS1 is located at P700, 700 nanometer wavelength. So here in non-cyclic photophosphorylation, you will be seeing the electrons which are excited in this reaction center of PS2. This is PS2. It is getting jump taken by the primary electron acceptor, and you can see the plastoquinone, and you will see the B6F complex, that is the cytochrome complex, and you will be having the plastocyanin molecule. So the electron is getting transported. So we can say this one electron transportation is taking place here. Primary electron acceptor is going to transport from plastoquinone to cytochrome to plastocyanin, and finally make it reach the photosystem 1. So just I'll mention here, it is not there. This is photosystem 2, and this is photosystem 1. Okay, already it is written here. P700 means photosystem 1, P680 means photosystem 2. From second complex, it is reaching the first one, and then afterwards the electrons getting transported here. After the electron is getting transported, this electron is getting transported. Now you already know that PS1 also, here also the same situation simultaneously happening, that is trapping of the solar energy by the light harvesting complex. Chlorophyll is uh, taking the energy and becoming high energy state, and because of this the outermost a pair of electrons are getting excited and jumping, and these pair of electrons, when they are jumped, here also the same situation, primary electron acceptor molecule is there. From P700, the electron acceptor will be accepted by ferredoxin. Here, ferredoxin. Remember about this plastoquinone, cytochrome, plastoquinone, cytochrome, plastocyanin will be there here. But here, when it comes to PS1, you are having only the ferredoxin is the electron acceptor, and what is happening, we'll see. Now, when the electron is moving, it is losing the energy, it is downhill. When it is moving from uh, photosystem 2, it is going downhill, down, and here you will be seeing that meanwhile the energy is given out. When the energy is given out, ATP synthesis is taking place. ATP synthesis is taking place because here ADP will be present, and inorganic phosphate they are going to get combined, and ATP synthesis will take place here. And here it is different. The electron which is given by this PS2 is taken by PS1. After taking that, why it will take? It will take because it is also losing its electron. Okay, whenever it will lose electron, again it will get back the electron. So here, this PS2's electron which is coming here will be taken by PS1, because this chlorophyll molecule is also losing the electron. That electron is going here like this to the ferredoxin, and it is given to NADP, which is an electron acceptor, and here enzyme NADP reductase enzyme is present, which is going to make the NADP accept the electron as well as the protons also. You can see H is nothing but the proton, and it is going to get reduced to NADPH. In this manner, ATP synthesis will take place, NADPH synthesis will take place, which are required in the Dock reaction. And the most important thing you have to learn here in this session, in this non-cyclic photophosphorylation, is about photolysis of water. You see here, two molecules of water, that means 4 protons and 4 electrons will be there, and oxygen will be there. And can you see this arrow mark? In this arrow mark, you are seeing electron is getting added here. That means when the splitting of water molecule is taking place, this water molecule will be having 4 protons. 2 molecules of water means 4 protons, 4 electrons, and O2, oxygen. So these 4 protons are there. You can see the 4 protons. Oxygen is there. Oxygen is liberated out. Okay, it is given out. Where these protons are moving, these protons are not going anywhere. They are going to this NADP, and here you can say this reduction is possible by the addition of these hydrogen protons and these electrons. So these electrons and these protons are taken up by this NADP and getting reduced to NADPH in the presence of enzyme called NADPH, NADP reductase enzyme. So you have to remember the enzyme name also. NADP reductase is the enzyme. So this water complex or photolysis complex is nowhere. It is in the grana only. It is located inside membrane. So this is the grana, outer membrane, inner membrane. So inside only, here only it is located, this photolysis complex, and it is splitting. How this water is getting split? By the energy of this chlorophyll only. It is splitting because this chlorophyll has lost its electron. 2 electrons jumped here. So it wants that electron back. So it cannot take back here. The electron is going like this and going to the PS1, and PS1 is also losing its electron, and that electron is taken by NADP and becoming NADPH. So you can see here in this fashion the electron is transported, and it is not coming back. The electron is not coming back to the chlorophyll. This chlorophyll's electron is given to this chlorophyll, and here this water molecule electron will be taken by this chlorophyll molecule. So the electron is not coming back. So it is called non-cyclic. Non-cyclic, and this is a photophosphorylation taking place. This photophosphorylation is meant for ATP synthesis, NADPH synthesis. I hope you are done with this, and we'll move to next. Electron transport. In electron transport, you can see photosystem 2, the reaction center chlorophyll A absorbs electron transport. Just now whatever I have discussed, the same thing is written here. In the PS2, the reaction center chlorophyll A absorbs 680 nanometer wavelength of red light, causing electrons to become excited and jump into an orbit farther from the atomic nucleus. And these electrons are picked up by an electron acceptor, which passes them to an electron transport system consisting of the cytochromes. And this movement of electrons from pheophytin to P700 is downhill in terms of oxidation reduction or redox potential. So this is what happening. This is a PS2, and from PS2 you can see electron jumping, and through this electron transport you can see the electron transport is taking place. Next, the electrons are not used up. Here the electrons are not utilized. They are getting transported only as they pass through the electron transport chain, but are passed on to the pigments of photosystem 1. The PS1 is going to receive that one. And you can see here in this picture also, just now I discussed clearly, this one PS2's electron is going like this and electron transport is done, and here in the PS1 is taking the electrons. It is taking electrons. Meanwhile, here also the same situation. Electrons will jump due to high energy state of this chlorophyll A, and it is going to be taken by primary electron acceptor. And here also it is passing through only ferridoxin. It is passing through only ferridoxin, and it is given to NADP. Okay. So here NADP is going to get reduced to NADPH in the presence of NADP reductase enzyme, and it also receives the hydrogens from the water molecule. Splitting of water molecules will give protons, electrons, and oxygen. These protons, hydrogen will be taken here to NADP, and the electrons of this water molecule is taken to the PS2. Simultaneously, electrons in the reaction center of PS1 are also excited when they received red light of wavelength 700 nanometer here, and are transferred to another acceptor molecule that has a greater redox potential, ferridoxin here. These electrons are then moved downhill again, this time to a molecule of energy rich NADP to NADPH reduced. Okay, so this is about electron transport. And further, in clear cut way, in this sentence it is clear that this whole scheme of transfer of electron starting from the PS2, that is P680, uphill to the acceptor, down the electron transport chain to PS1, excitation of electrons, transferred to another acceptor, and finally downhill to NADP. Finally downhill to NADP here, causing it to be reduced to NADPH is called Z scheme. As you can see, we can see here, the electrons from here is going to the primary electron acceptor. The electron transport system is done and it is reaching the PS1. And from here also PS1's electron is also getting excited and jumping and excited, and it is taken by primary electron acceptor and given to ferridoxin and finally to the NADPH reduce. So here we are getting a Z shape here, and that's why the name Z scheme. So causing it to be reduced to NADPH is called Z scheme due to its characteristic shape. This shape is formed when all the carriers are placed in a sequence on a redox potential scale. So on a redox potential scale, reduction and oxidation. Redox means nothing but reduction and oxygen. Recall once again in your earlier classes, primary 10th class, you have learned about oxidation and reduction. One shortcut is Leo and Gar. Leo means lion, Gar means it will make the sound Gar. Leo, loss of electron is called oxidation. Whenever electrons are lost, it is called oxidation. Gain of electron is called reduction. Gain of electron, when it will happen, when anything is losing only, no. So whenever oxidation is taking place, reduction is also taking place. Whenever loss of electron is done, any substance is losing loss of electron, it is going to get oxidized. And whenever any other substance, electron acceptors are gaining electrons, it is called reduction. Gain of electron is called reduction. So this is the simple funda about this redox, how it is taking place. And next here, for a clarity, non-cyclic photophosphorylation, you have seen PEA. PEA nothing but here you can see in this the primary electron acceptor P680 electron acceptor is taken and it is given to PQ plastoquinone, and BFC complex is nothing but cytochrome, and PC is plastocyanin. It is given to PS1 700, and PS1 at 700 wavelength also sunlight energy is trapped and electron get excited by the chlorophyll A molecule. It is also taken by PEA primary electron acceptor here and given it to FD ferridoxin, and ferridoxin finally gives it to NADP. So meanwhile, the most important thing you have to remember in this is ATP synthesis is taking place. When the electron is moving here, the energy of that electron is lost and this energy is utilized for synthesis of ATP.